My name is Meredith Lee, and this is my protein presentation for Cell Bio 2440, and my protein is deoxyribonuclease. So I'm going to start by explaining proteins. So proteins are the building blocks that assemble cells and provide cells with their shape, structure, as well as being an integral part of executing the cell's function. Proteins have many different functions based on their shape. Some of the many functions proteins have would be catalyzing covalent bond with breakage or formation, provide support to cells and tissue, transports small molecules or ions, generates movement in cells and tissues, store small molecules or ions, signal carriers between cells, detect signals and transmit them to the cell's response mechanisms, and regulate genes. So proteins have a 3D structure, and there's 4 levels of the protein structure. You have your primary structure, which is the amino acid sequence of the protein. You also have your secondary structure, which includes the alpha helices and the beta sheets. Then you have your tertiary structure, which is the full 3D conformation of entire polypeptide chain, including the alpha helices and beta sheets. Then you have your quaternary structure, which is only provided when the protein is made up of more than one polypeptide chain. So now on to talking about deoxyribonuclease. It's a nucleic acid hydrolyzing enzyme. Its main function is hydrolyzing DNA in the presence of bivalent cations to produce 5 mononucleotides. So the bivalent cations would be, an example would be magnesium 2+. You have your different types. The 2 main common are DNase 1 and DNase 2, and I focused on DNase 1. So it's the most studied. It's optimal at neutral pH, requires bivalent metal ions for catalytic activity, and forms oligonucleotides with phosphate at 5' end. Major extracellular endonuclease secreted by digestive glands, lacrimal glands, and renal proximal tubular cells, and then it cleaves double-stranded DNA. So it's also located, it's mostly in like the digestive tract and places like that, but it's in cells. It's an extracellular enzyme, but it's located in all organisms of different species, and it's and it's a big one is in the pancreas, or that's where it's mostly located. Then you have your the DNase 1 structure. So it contains 5 amino acid residues, has 2 disulfide bonds with N-terminal signal peptide. Its secondary structure includes 2 tightly packed beta sheets, 8 alpha helices, and several loops. And this secondary structure is responsible for its rigid structure and structural stability. So this is my ribbon model. As you can see, the yellow is the beta sheets, and then the hot pink and then the dark purple color at the bottom left are the alpha helices, and it's my figure 1 for the purposes of the references. And then this is the gene, DNase 1 gene. It was discovered in 1995. It's about 3.2 kb long. It's composed of 9 exons separated by 8 introns. And it's figure 2. And here are my references.